Now I'm going to demonstrate that we can actually export any ROI at any resolution of our histology stack to a NIfTI image. So say I want to export a NIfTI image of just this little section of hippocampus showing the pyramidal cell layer and some of the granule cell layer surrounding the dentate gyrus. Okay, so again, same thing, export slide stack. This time I'm going to just export the first slide, so let's say slide range to export only the visible slides. And view area to export, this time let's choose the current view window, current window view. So you'll see that the text boxes here have updated to match only the current view window. Let's choose to export quite a high resolution image. Let's export something at a 2000 pixels wide, preserving the aspect ratio, and let's export it as a color RGB NIfTI image. Let's call this test stack 2. And let's open it up in ITK Snap. So here's that slice. You'll see that because we checked account for variable thickness and spacing, there is this additional slice before the slide that we want. But here is our NIfTI image of the histology exported at full resolution. Very handy tool for exporting your slide stacks to collaborators. We can also export segmentation. So I'll go back to the project and draw a little segmentation region here. Oh, that's bad. Let's do it again. Let's delete those. Do it again. Let's highlight just the granule cell, or just the pyramidal cell layer here in red, and the granule cell layer down here in orange with the second label. And this time, let's go export slide stack. Let's uncheck account for variable slice spacing, and say export segmentation labels as well. And let's just overwrite that test stack and replace it. Now it's asking us to export the segmentation stack as a 3D volume. So this is the test stack, but these are the segmentation labels. So let's close Snap and open up the stack. So here's our image. And now the segmentation labels have been rasterized and converted to NIfTI as well, and they can be loaded on top of our NIfTI image of the slice. You'll notice that label 1, here's label 1, label 2. These correspond to the ordering of the labels in our list of IDs. So even though label 2 is orange in HistoloZee, and label 2 is green in ITK Snap, you can be assured that they're the same segmentation label because they have the same ID. Now, this concept can be extended to much more complex projects. Let's load up a reconstruction of the entire slide stack now, which has been exported to NIfTI. So I did this ahead of time. I have reconstructed our slide stack and saved it as a NIfTI image in RGB format. Here is the entire slide stack now exported as a three-dimensional NIfTI image with 3D world coordinates preserved. and you can view histology in 3 dimensions now. And since world coordinates have been preserved, we can load up the corresponding MRI in another snap window, and if all goes well, we should be able to link the cursors between these 2 windows without a problem. So there it is. Through the HistoloZee reconstruction pipeline, we have mapped a stack of histology slides interactively to world coordinates that correspond to the space of our MRI image. Now, the alignment here isn't perfect, but it's just to demonstrate that this can be done. And so in theory, you could go through your MRI here, inspect, say, this dark layer, and actually find out what it corresponds to histologically using various immunohistochemical stains. Or you could go here to the pyramidal layer and see exactly what kinds of cells are there. And everything is 3D, so you can view histology in 3 dimensions as well. You'll notice that spacings have been inserted using blank slices without compromising the alignment to world coordinates. And extending this further, we can actually show alignments between our whole histology block, our whole tissue specimen, and all the blocks. So this now incorporates some warping out of plane. This is a reconstruction of all histology samples now to all the blocks warped into the space of the whole MRI image, not just the block MRI images. So if we load up the whole MRI image here, as an overlay, you can see that histology has been aligned to the whole MRI. I skipped quite a few steps in this last video just to show you the possibilities of what can be done using this software.